Hello there, welcome back. So this question is on photosynthesis, still topic 5. So it says scientists studied the rate of carbon dioxide uptake by grape plant leaves. Grape leaves have stomata on them. Take note of that word stomata on the lower surface, but not stomata on the upper surface. So the scientists recorded the carbon dioxide uptake by grape leaves with 3 different treatment. Treatment 1, no air sealing grease was applied. So at this point, please pause the video and see if you can just read through the questions, okay? And like I always say, try to attempt the question before seeing the answer, and preferably make sure you've read this topic. Now treat the lower surface of the leaf. The lower surface of the leaf was covered in air sealing grease that prevent gas exchange. So both lower surface and upper surface of the leaf were covered with the air sealing grease that prevent gas exchange. The scientists then measured the rate of carbon dioxide uptake by each leaf for 60 minutes in the light and then for 20 minutes in the dark. The result of the scientists is shown below. So treatment 1, treatment 2, treatment 3. So you might I think this is okay. I wanted you to say you should want the video to see the upper part. So treatment 1, 2, 3, you can see each of this line represents that. This one, treatment 3, treatment 2, treatment 1, and set well. So try to pause this video at this point and see if you can just study that for the moment. Okay, I haven't established that. It says suggest the purpose of each of the 3 leaf treatment. Why was there no sealing here? Where was sealing on the surface here only? Where was there sealing on both surfaces here? Okay, attempt that. So the reason is no grease measures. For the first one, no grease means stomata are open, thereby allowing the normal gas exchange or normal uptake of carbon dioxide. The one that has it under the surface, grease on the lower surface, means that stomata are sealed, thereby preventing the uptake of the carbon dioxide. Okay, then treatment 3, grease on both surfaces, thereby both surfaces are sealed, making carbon dioxide uptake impossible. So basically this one is almost the same, but remember this one's more like a control because no surface is sealed and all of that. Okay, I haven't established that. Let's proceed on this. Now describe the results shown for treatment 1. So if you look at treatment 1, I would probably want you to go back here. So look at this and this, because this is treatment 1. Describe that. You can see the uptake of carbon dioxide. And I always say that questions like this usually should be easier for students to get because it's something you can describe. It's more like just describe what you're saying in time and all of that. Okay, let's see. Describe Tumaka. Okay. Now it says that this means the rate of carbon dioxide uptake was constant at 4.5 until. What does that even mean? If I go back, this particular document, you will need it later on. That's why I wrote it somewhere there. Okay, so you say it was constant here, then around 60 something minutes or there about 70 minutes it drops. So can you see it was constant until the light was turned off and then fell to 0? Like it just, I think I should keep it there. Okay, I don't know if I can keep it together. No. Seriously, I can't even see this document anymore. Okay. Okay, that's quite not the way I wanted it to be. Okay, let's do it this way. Okay, I think this might be helpful. Yeah, I don't want you guys to be able to see the question simultaneously, but that was becoming somehow. Okay, so I think this way you should be able to get to see that. Yeah, okay, I think this is better. Yeah, this is better. So it says this means that the rate of carbon dioxide uptake was constant until light was turned off. So when you look at this, when the light was turned off, then it dropped. So when uptake fell from this to zero, it started to fall to 60 minutes, then reached lowest around 80 minutes. So it means at this point, when it was 60 minutes, that's when it was dropping, then it totally dropped, totally like stopped. So it was reducing. Uptake was beginning to drop. Like it didn't just stop to zero. It was when it was 60 at 4.5, the light was turned off, then it was dropping gradually till it gets to. So it was actually until 80 minutes before it dropped to zero. So it wasn't just like that. So we said it fall to it, but it wasn't until 80 minutes, really, if you get what I mean. So it was turned off here and it started dropping, took some time. So it's like it took 20 minutes interval before it finally dropped to zero, but it was dropping. It was dropping. Okay, that's why it wasn't. If it dropped to zero directly, it should have just come straight down this way. Okay. Now the next question says this. Oh no, stomata was. Is that the second one? Stomata was closed when the light is turned off. Explain the advantage of this to the plant. The stomata closed when the light was turned off. What is the advantage of that? Why should that be advantageous? Now, it prevents water loss. If you remember, if you have done transpiration, it prevents water loss. So as stomata closes in dim light, this helps to maintain water content. You remember that stomata is the same opening through which transpiration takes place. So if the stomata is opened at all times, that means water will be lost at all times. And this what happens when you are carrying out photosynthesis. Carbon dioxide is coming in, oxygen is going out, as well as water. So now in dim light, when there's no more sunlight, there's no more photosynthesis really taking place, when the stomata now seals, that means water is not going out, oxygen too will not be going out that much. Please, it doesn't mean it will not go out at all. It's going to go out a little. And as a matter of fact, let me say this: even in the dark, anyways, the cells of the plants will be carrying out respiration. So oxygen will not even be going out. Rather, it will be coming in, or the ones already in the cell. But bear in mind that it is going to be, if I thought it is open, only few will be opened, really. So, but most times, stomata will be closed for some period, so that water loss is reduced. Okay. The reason why I have to explain this now is because so that will probably feel like, okay, what happens to oxygen? Because at night, oxygen is coming to the plant, and if you say stomata is totally closed, when will they have oxygen to come in to carry out respiration? So respiration will still happen, okay, the stomata opens little, and not just open little, there's some oxygen already trapped in the cell which will be used to carry out respiration. Okay, but for the purpose of this question, this is what it will be, and that's really what it is in reality. Okay, so having established that, I'm trying to get that off. Next question says, Treatment 2 shows that even when the lower surface of the leaf is sealed, there is still some uptake of carbon dioxide. Let's check that out. So treatment 2, let's treatment 2, yes. So because according to what we said, treatment 2, lower surface was being sealed. So according to that, even though it was still sealed, because from the beginning it's still sealed and there's still carbon dioxide being uptake, even though it's just some, it's not up to 1, but it's still happening. So excuse me, what does the question say? Suggest how this uptake continues. Yeah, even though it has been sealed. Why? The leaf was able to take in carbon dioxide through its upper surface. So that's why you saw that for the one that is number 3, which was sealed on both surfaces, there was nothing. But because this one was only sealed on other surface, it was still able to take up some carbon dioxide through its upper surface. Hope that made sense. Now, in treatment 1 and 2, the uptake, I hope you are saying that clearly, the uptake of carbon dioxide falls to 0 when the light was turned off. Why? Well, that should be direct. Try to see if you can answer that. The reason is simple. In the dark, there is no use of carbon dioxide for photosynthesis. So, because since there's no carbon dioxide coming in, or let me say, yeah, because in the dark you don't have it's light independent stage, and what we need there, what is happening is carving cycle, is whatever has entered into the leaf, that's what is happening. So no diffusion gradient is maintained for carbon dioxide to take place. So don't forget the fact that it says that, don't forget the fact that in the dark there's no carbon dioxide coming in. No, because it's not needed. What is coming in, if at anything is coming in really, will be oxygen for respiration. Don't forget that all this equation we normally write C6H12 O6 plus I'm literally writing respiration. It's meant to be CO2 plus H2O gives you C6H12 plus 6CO2. 6O2, 6 and 6. Now please bear this in mind. So this is photosynthesis, right? Now this is happening in the day. At night, this goes here and this comes here, which means oxygen is now coming in to combine with this at night. Please don't forget that, because respiration basically, photosynthesis, if you flip this equation, the opposite will now be aerobic respiration. Why I write this was just to show you the fact that oxygen is what one that is coming in in the dark or during light independent reaction. Hope that made sense. Next question. It says that a student investigated the effect of different wavelengths of light on the rate of photosynthesis. She used the apparatus shown in figure 1. So we can say light of a particular wavelength, suspension of photosynthesizing agar gel in the oxygen meter. Okay, so sorry, I'm saying two things together. So light of a particular wavelength is there, suspension of photosynthesizing agar cell is there, then you can see oxygen meter put there. So the question is, what measurement should the student have taken to determine the rate of photosynthesis? The rate, take note of that word, the rate. Well, it should use how much of oxygen is being produced. So the faster the oxygen produced, the more we can know that photosynthesis is taking place. So when you see the oxygen meter there, is to show us, okay, oxygen is coming out. Okay, so for real, that means the algal cells are actually photosynthesizing. Okay, the next one says, what measurement, other than temperature and pH, give two factors which should be kept constant during this investigation. What other things should be kept constant? The light intensity, so yeah, it shouldn't fluctuate. Then the amount of algae cell in the suspension, so the amount shouldn't be reducing it or increasing it, because that will affect the results. Okay, so also the light intensity, if you are increasing the light intensity, you should expect that there will be more photosynthesis, but it should be kept constant. Now, the student did not use a buffer to maintain the pH of the solution. Explain what will happen to the pH of the solution during this investigation. So he's saying here, buffer is to either, if there's too much acidity, it will neutralize acidity. If there's too much alkalinity, it will neutralize alkalinity. But here, from what you are seeing here, the student has not added any buffer. So what do you think will happen eventually in this investigation? What would happen with this setup? How many markers? 2 markers. Okay. Now, sorry, I didn't finish showing you this. So other things that can be kept constant will be this: same species of algae, the time, the level of carbon dioxide, and concentration. These are the other things that can be kept constant, or that should be kept constant. So peradventure, maybe one of the things you wrote when you were answering this question was not these two. Either of these two is also correct. Okay, the time, level of carbon dioxide, concentration should be kept constant. All of those, so to say. Now back to the question we're actually on at the moment. It says the pH will increase. Why? Because over time, carbon dioxide will be used up for photosynthesis. So, and once that happens, the pH will increase, because as the whole thing continues, carbon dioxide will be used up, will be in this setup. So once that happens, the pH will end up increasing. So it already said, the first sentence already said the pH will increase. Okay. Okay. Now, figure 2 shows the student's results. So we can see here rate of photosynthesis. Here, this is wavelength. So suggest and explain why the rate of photosynthesis was low at 525 nanometer and 575 nanometer wavelength. So it was low around here and just here, yeah, between there, basically around there. Yeah, why was it low there? Well, if you look at the wavelength and timing. Now let's look at this. Yeah, just the way I circled it. This is because less light is absorbed at this stage. At this wavelength, green light is reflected. Hence, light is available for photosynthesis at the light dependent stage, so to say. So less light is available. Now you know that green light is really what we need. I mean, like, green wavelength is what you need. So at that wavelength, the light is more reflected. So because it's more reflected, the amount of light absorbed is now lesser, and for that reason, less light is now available for photosynthesis, for the light dependent stage. You know that light dependent stage is when we actually need light. So if it is during the dark stage, nobody needs light. Whether there's light or not, that one will happen. So because of that wavelength is counterproductive to the light dependent reaction stage. Okay, now this is the part where I really want you to really pay close attention here. It's quite some serious question. Now, so this is 6 marker, 4 marker, 5 marker. Now, the reason why you probably, so you see the space here is quite tight. That's why I have this one here. Probably you saw it pop up sometimes ago. Now, on island in the Caribbean, there are almost 1,005 species of lizard belonging to the genus Anolis. Scientists believe that these species evolved from 2 species found on mainland USA. Explain how the Caribbean species could have evolved, please. Okay, that's the question. About 150 species are there. This kind of question, you need to first pause the video here and think. Okay, pause the video here and see and think about it. So for me, you see, I have to refer to document tagged answer to question 3 of photosynthesis. So I literally had to create that here because there was not enough space. So try to see. So all of the answers to these ones are basically in this document. So pause the video at this point and see if you can attempt that. If you have done that, let's have a go together on this. So question 3, a new species can evolve due to geographical isolation over time. Okay, when this happen, there will be no gene flow. Mind you, sorry, this particular question really is not really meant to be in this topic 5, really. Yeah, but for some reason, the whoever compiled this file did that. Sorry about that. Okay. Now, so when this happens, there will be no gene flow between the population and hence a separation of gene pool. Now, different abiotic and abiotic pressure are now and environmental factors will exert different selection pressure. So, since there's that geographical separation between a population or a particular species, then you should know that there will be, even though they were supposed to be new species, the same species before together, but because of the geographical separation, there won't be gene flow amongst the population anymore. For that reason, the gene pool will now be separate. Then they will now have different abiotic factors, even biotic factors upon them. The weather might have changed. The process of looking for food might have changed. Yet, so basically, the environmental factor and selection pressure will now be different. So over time, there's a random mutation in the separated species, leading to what? Variation. Now, because there's now selection pressure that probably led to mutation. Then once there's mutation, change in gene. Once there's change in gene, there will be change in structure, which is basically variation. So with time, don't forget this is a 6 marker. With time, the species evolve different reproductive structure and reproductive behavior. As such, only species with compatible reproductive rituals can survive and reproduce. This then leads to change and increase in alleles frequency in that species. Now, in the new species. So the newly evolved characteristics is passed on to the offspring over many generations. Now, hear this. You might not, any question that has to do with evolution, natural selection, and the likes is the same pattern. What you want to do is you want to first talk about the fact that maybe there's a separation. Try to follow the question prompt. There's an isolation. If it's an isolation, or sometimes there's no isolation, the question might make you talk about the fact that there's the reproductive structures does not just match anymore. Their rituals are not matching anymore. But what you just want to drive home is the fact that there will be this part must be constant. The fact that there's a new there's a variation. The variation can be caused. The question might give you might have said it what the variation was already, or it is caused by random mutation, but there's a variation. And once there's a variation, then that will lead to they won't be able to stay together anymore. As such, there will be In this case, reproductive rituals cannot match anymore. And the most important part that must be present is this part here. In any of the things you're writing about evolution and natural selection, things like that, you must always remember to add over many generations. It's not something that just happened within 2 generations. It has to be many generations before you can have a new species, because evolution usually takes maybe thousands of years to happen. Not maybe thousands of years to happen. Okay? The next question says, the Anodis segri is a species of lizard that is found on, or is it sagria? Don't worry, I just pronounce it in a way, on some of the smallest Caribbean islands. Describe how you could use the mark release capture method to estimate the number of this. So you should know that from your population study and all of those things. So the mark and release, that's what's basically happening. This is not an applied question. It's just, if you know it, you know it. So there you go. So it says here that the lizard is captured and some of these are marked and released. The method of marking does not harm. That's something you have to know. You would have learned that at school, that whatever you're going to use should not harm the organism. It should not make them, see the next one, it should not make them more visible to predators. Basically, like, they should not be too visible to predators to see them. So the marking used is not easily erased. So that's quite a funny one. It's not too, you can imagine that it should not be visible, and at the same time it should not be easily erased. Well, the truth is, it doesn't have to be bright for it to stick better. That there should be sufficient time. Sufficient time should be allowed to make the lizard randomly distribute among the island before the second sample, before you take them again, before you went and go and catch the population again. So there should be enough time, so to say. So you can pause the video here to read through all of this. Now, lastly, most importantly, the population is calculated by what? Number in the first sample that you first took, multiplied by the number in the second sample, divided by the number of recaptured. So as many as you can, when you capture them again, as many as you can see the mark on. But this one, usually you might not have, you might do it. Some of you must have done it at school. You should have done it at school anyways. But the number you can recapture will be divided by that. So it's just number to calculate the population, number in the first sample multiplied by number of second sample divided by those that you recapture, basically. So that is how you calculate that, or that is how you do that investigation, because this is eventually, when you follow all of these proceedings, this is the last you're meant to do. Then okay, the last question there says, large area of tropical forests are still found on some Caribbean. The concentration of carbon dioxide in the air of the forest changes over time, over a period of 24 hours, and at different heights above the ground. That's quite interesting. Use your knowledge of photosynthesis and respiration to describe and explain how the concentration of carbon dioxide in the air changes. So it's like it changes from level to level like that. So over a period of 24 hours and a different height above the ground. Why does it change with time? Why does it changes with different level, like strata? Like, yeah, why does it changes? That's quite interesting. Now, the fluctuation is based on weather. So photosynthesis or respiration is dominant or not. So if it is in the morning, you should expect that there will be more photosynthesis. If it's at night, you should know that there will be more respiration, like I said in the previous question. At night, the concentration of carbon dioxide is higher as more carbon dioxide is being produced via respiration with no photosynthesis in the dark. So you get what I'm saying? So during the day, the concentration of CO2 is lower as there's a lot of of it being used during photosynthesis. Can you see that? So this is based on time. This is based on time. Then based on height, like different levels. Concentration of carbon dioxide decreases as more CO2 is absorbed with increase in height. Okay, so as you move up, it's expected to be absorbed higher at higher level. On the ground, however, there is higher concentration of carbon dioxide due to less photosynthesis and more respiration. So it means that the higher you go, the more you will see the more carbon dioxide will be absorbed in the forest. Yes, basically. And then that should even be easier to understand because it's easier for wind to blow and all of those things, so to say, up there compared to the higher you go, the colder it becomes because it's more windy. Okay, hope that made sense. Farm land previously used for growing crops was left for 30 years and developed into woodland. During this period, ecologists recorded an increase in diversity of plants, of birds in the area. Name the process that resulted in development of woodland from farmland. That's just one marker. Sorry, this question might not be, it's not really a photosynthesis kind of question, but it's okay. What happened in succession? Explain the increase in diversity of birds as the woodland develops. 3 markers. There will be a different species of plants or different plants which will attract different insect species. Now, see the trend, because please, things don't just happen in habitats. There must be something that will cause something to happen. So there must be some different plants that will make some insects to grow on them, and for that insect, so it says more insects will attract different animals such as birds. So different insects, different birds. This increases variety of food for different animal species. So then this will lead to variety of habitat as well as different niche. Niche means the role an organism performs. So you basically have different birds, different some will be eating on, some will be seed eaters based on the plants, some will eat leaves, some different things, so to say. So some birds probably may even eat other birds, yes, so to say. So but you saw that it is from diversity of birds in the woodland. So it didn't start by just talking about the birds. You have to first refer to the fact to the plant, from plant to smaller plant, just the way sometimes students don't know that some of the understanding here comes from food chain. Producers first, you have different producers, you have different consumers, period. That's what is happening here. Okay, so the ecologists also investigated photosynthesis in 2 species of plants found in the woodland. One of the species was adapted to growing in bright sunlight, and the other was adapted to growing in the shade. So the ecologists' result are shown. So this kind of question, you pause the video first. Pause the video. So give 2 factors which could be limiting the rate of photosynthesis in the sun. In the sun plants between point A and point B. So here, this is some plant between here. What might be the limiting factor that probably made this to now remain constant, so to say? Take note of all of this and all of this. So the limiting factor will be temperature here, because when other things, temperature and carbon dioxide concentration. So that's because of this light intensity in a way refers to temperature and also the carbon dioxide concentration, basically. So okay, then explain why CO2 uptake is a measure of net productivity. Why is CO2 that it shows gross photosynthesis in a plant. So like it shows how much photosynthesis is happening in a plant. Now, how do we calculate that in what's that word? How do we calculate that by virtue of mathematical thing? Net productivity is always gross photosynthesis take away respiration, basically. I think I did that also in energy and ecosystem. So I think I'm trying to think there's a question like that on that has to do with this on that. Now use information in the figure to explain how the shade plant is better adapted than the sun plant in growing at low light intensity. If you look at this, so how is this one more better adapted compared to this? Really? Okay. Under the shade, the plant has lesser respiration, hence lesser CO2 is released. This makes more CO2 available for sugar production, which then increase productivity. That's